All right, so today, guys, let me just tell you something. I really didn't expect to see so many people so happy. Okay, thank you for being here. And I do think the recording's going to work today, so that's going to be a great thing for the people that are out. And there are a few people that are out. What I'd like to do is give, without, since a lot of counties were closed, and this is the way this is going to happen, okay? If the college is open, I'm employed to be here and holding classes. So I will be here having class, even if it's 3 people here. But I do not want to count against you if your counties were closed. It means that they thought the roads were dangerous enough that they didn't think people should be out on them. So I don't want it to hurt you if you don't come in because your counties were closed, but I do want to give you all credit. So it's not going to hurt the people if you're listening. This is not going to hurt you that you're not here, but the people who are here are going to be given a little bit of credit for being here today. And what I'd like you to do is use your cover sheet. And a cover sheet, just a blank piece of paper with no notes on it, whatever. And Michelle, if you would like to, you can do it from the previous lecture since you just hearing this one, okay? But what I'd like you to do is on your cover sheet, I would like for you to write down, you know, as long as there's 5 things. So I don't want this to take very long. To take a class, I'm mostly doing this just so I'll have your paper so I can give you participation for today for being here. The other people will just see a blank next to this date. It won't be hurting their grade at all, but this will be helping y'all's grade. So Brandy, what I'm saying is on your cover sheet that's blank, I would just like you to write down 5 facts that you found interesting. Just make them as a fact, a statement of fact, okay? So 5 facts that you found interesting from lecture, and that will be it. We'll turn those in. If y'all could just take about 5 minutes to do that, no more than that, that'd be great, okay? And again, Michelle, you can use from the previous lecture if you like. So just 5 statements, if you will, about things that you didn't know, but now you kind of know, or know more about maybe. You know, people who are coming with that kind of barrier. All right, so thanks for being here. And what I'd like to remind everybody is I did give out a handout in lab, which is just our little introduction to the microscopes. So you would be able to label those microscopes Wednesday and then answer 2 questions. Do you remember the 2 questions? One was, how do you determine the magnification of what you're looking at right then and there in that live time? How do you determine what the magnification is? What do you need to do? Exactly. That was the jumble. But it's multiplying the ocular lens magnification by whatever objective lens magnification you've got in use, right? And multiplying those 2 numbers gives you the total magnification. The second question we asked was, why is it important that you know what the magnification is of what you're looking at?
So it helps to identify
so you know what you're looking at. One word, identification. So you can use that to help you identify whatever you're looking at. So they were the 2 questions you want to be able to ask. So you guys will be ready for just that brief little microscope quiz by Wednesday, wouldn't you? Even if you're not here just listening in, you would be ready for that. Okay, I do want to make an announcement that typically what we're going to be doing is having our lab time on Mondays, and then that's usually typically going to be our lab time. And then on Wednesdays, every now and then we might just go in there and look at something that we set up on Monday, because it would be 48 hours later and we'd be needing to look at it, right? But the time that you really want to devote or commit to is going for lab is going to be on your after your Monday lecture, okay? And usually we'll be out much earlier on Wednesdays. Fair enough? Y'all can plan that? Michelle, I know you had been in the section that got canceled last week, I think, right? I hope you had not purchased the eScience code. And if you, if you haven't opened it, you can. So please do, okay? Because this class does not require any cost for lab, because we'll be doing things in lab. And I only had those as long as they allow me a budget, which I think they will enough. Okay. So thank you guys for being here. And like I said, this wasn't really a quiz, but it was just you letting me know that you had looked over it and wrote down some facts. Any questions for me before we get started though? Today? Yes, Jared? Well, so no, not really. No. So no. But Wednesday is going to be just a little thing, okay? So this week, but starting next week on Monday, we really will start with the Monday labs, right? And then the Wednesday sort of just finishing up if we have anything to finish up or look at or clean up. Yes.
Next Monday? That's what I was about.
What's going on next Monday?
Martin Luther King.
Okay. All right. See, I always forget holidays. Okay, so is the college closed on that day? Are you sure? Are you 100% sure?
Wow, look on sometime.
So, okay, does it say it in our syllabus? Because I would have put important dates at the end of that syllabus. Does it say that? Okay, so,
But I saw it on my last
we'll,
classes.
all right, well, we'll know it when, you know, I'll look it up before we leave today. Somebody will. Okay. All right, so, on what?
Online, I went online.
Online, it says the college is closed, Monday. If the college is closed, I'm not supposed to be here. So y'all don't have to be here because it's actually a legal kind of thing. I was going to pretend like I was home and I was going to yell down the hallway, "Are we open?" Next time, if I saw somebody out there, I was really going to do that. Okay, forgive me. All right. What'd you say?
I want to be like you.
You want me to go ask about it?
Yeah, Dylan's here. He's a guest today, so he can go do that. And like I said, if the college is ever closed, actually, there really are legal reasons that students aren't supposed to be here. So, I mean, we aren't supposed to be here. So, of course, we wouldn't have class Monday, and then I don't know. So maybe we'll start that lab Monday thing after that. Okay. All right. So what else? All right. Let's go ahead and get started with this. So, did anybody see anything in the news or anything they wanted to bring to attention to anybody? To everybody. There were a couple things, but I certainly can't remember right now. But maybe if they come to me, we'll sort of stop notes or whatever. Because I do want to hold this class not just by a textbook and just whatever. I want us to bring current events in, and if you all see something that you find interesting, bring it in so that we can discuss it. Certainly, if I would love that if you did that. Jared, did you have something else? Yeah.
It's not really a current event, but
Okay.
while I was in the grocery store the other day, I did see a organic drink that had bacteria as an ingredient.
That's awesome.
Various genus and species of them.
Yeah, so it had the genus and species name of the bacterium that was in this particular.
That was a
No, he's absolutely right. He's absolutely right. And does anybody know what that is when you hear about food that actually has contained in the food and it's advertised as something that's very good to have microorganisms in the food? Do you know what that's called? The probiotics. Is that you, Janet? Okay, probiotics. And actually, I want you all, that's great. So thanks, Jared, for bringing it up in class. We'll talk about it later in here as well. But probiotics, when you hear this advertised in food and whatnot, and you'll hopefully, now that you've sort of been tuned into that word, you'll be hearing it a lot and seeing it. But probiotics is the idea that we need microorganisms in our guts, in our colons, and in our when I say guts, it sounds whatever, but that's really true. It is. We need it in our GI tract, don't we? We need them on our skin. We need them in our vaginas, ladies. We need them at certain proportions, if you will. And what happens now in our world is that we have a lot of foods with preservatives and high salt content and all kinds of chemicals and dyes that have been added and trans fats and that kind of stuff. what do you think that those things do to the normal flora that are in our guts, if you will, and our colons especially? What do you think that does to them? Well, it doesn't really destroy it because it's hard to, it's hard to become sterile, if you will, but it definitely changes the microorganisms in our environment. And so what happens is that's so understood that they are adding microbes to our foods and using that as an advertisement, as a healthy thing and a good thing. Now, the problem, and it's not really a problem, it's not a problem, let me take that word back. Are we closed?
Yes,
Okay, so no class Monday.
and the Wi-Fi network is down right now too, so if anybody tries to log on, probably not gonna work.
Okay.
Lucky you.
Yeah. Okay, so they wouldn't do that in my class anyway, John, right?
What's
My
that?
class, no electronic devices, right? Except for you when you're doing your work. Okay. All right, so anyway, the thing about probiotics, they're new enough that we're not really sure how effective they are yet. And so there's a lot of research going on right now to figure out, but it is really highly recommended that anytime anyone goes on an antibiotic, I mean for anything, a urinary tract infection, strep throat, a sinus infection, which you shouldn't have been treated with an antibiotic, but okay. Anytime anybody goes on an antibiotic for anything, it is highly recommended that they go ahead and take a probiotic. And they try to include foods that are enriched with probiotics. So that is, they don't hurt you, and we're hoping that they help you. How are we hoping that they help you? By reestablishing normal flora.
You said you
Yes,
need,
Brandy.
you said you need or have to be on a probiotic?
You don't have to. It's highly recommended that you do take one. If you're taking an antibiotic for any reason, it is highly recommended that you do. Most physicians now are recommending probiotics when they give a prescription for an antibiotic. But here's the deal. We should probably be trying to take them anyway, and if we can. Some of them are kind of expensive, but certainly eating foods that have been, that enhanced with probiotics might not be a bad idea. We just don't know how effective they really are. The best thing that we can do is eat plenty of fruits and vegetables that we haven't steamed to death, cooked to death, or put so much salt in them that we killed everything in them, and you all know how we cook vegetables sometimes, especially in the deep south, right? It's not good unless it's completely unrecognizable as to how it came off the vine and it's got a lot of salt added to it. Now that just sort of messed up. What'd you say? Mac and cheese is not a vegetable. I know, it should be those. So anyway, but thank you for bringing that up. Absolutely important is understanding probiotics. And we do know we can get them in nature if we just go out and dig up a potato, wipe it off on whatever we're wearing, and just eat into it. Go ahead. Just not overcook our vegetables and eat enough fruits, right? Then we're getting them kind of naturally. Yogurts, cheeses, those types of things are rich in those. So that's a very good thing to bring up. And it seemed like you brought up somebody brought up something else I want to say, but we might get back to it. Anybody else? I thought this is what I thought you were going to say. I thought you were going to say with this government shutdown that the inspectors are not there looking at taking samples of our fruits and vegetables and all the foods in the grocery stores right now, getting samples so that we can see when there might be outbreaks of things that aren't probiotics, but things that are actually disease causing, like E. coli. Y'all have all heard of E. coli, haven't you? E. coli, C-O-L-I. The E stands for, and I would expect you to look this up, Escherichia. Escherichia is the genus, Escherichia coli. It's a bacterium that is not the human E. coli. It's a subspecies. Just write E. coli for now. But that's found in all animals' colons. But the E. coli subspecies that are found in like cows and other animals can be very harmful to us. coli, C-O-L-I. E is the genus. E stands for Escherichia. Coli, C-O-L-I, is the species. And you will hear about E. coli outbreaks. How many of you have heard of E. coli outbreaks? Yeah, and so, but we have usually the FDA and their workers are making sure they're sampling these things so that we don't hear about them too often, do it. Can they kill people?
Yes.
Yes, they can. And then we also hear about, I'll go ahead and add another organism to your list, these microorganisms to your list. And by the way, I see on some of y'all's little study reviews that you're not really caring too much about looking things up, how to spell them. Please, please, I might start counting off on you for that. I am definitely may shame you into it, okay? So please, please don't put things that are misspelled, like the Mycobacterium avium intracellulare. That was all kinds of craziness. I saw about that one. Y'all look these things up, okay? And make sure you're not spelling them incorrectly for your friends. Okay, so here's another one though. I will give you this one. The genus is called Listeria. Listeria. And I will spell it just the genus. Listeria, Listeria. That's the genus. The species is Monocytogenes, but you do not need to put that right now. Just put Listeria genus. This is one that is very, very common that can cause outbreaks. And it really has a significant mortality rate. When I say mortality rate, what do I mean? It means of the people that get sick, how many do you expect to die? That's the mortality rate. So every disease that we know and we've studied has a particular mortality rate associated with it. So Listeria is found in a lot of meat products, especially deli meats and some vegetables. So now that we don't have these inspectors sampling, do we expect to see more outbreaks during this period of time? We really do. So this whole government shutdown is not just affecting the people who are not getting paychecks, though that breaks my heart for them, but it's affecting everyone. So even if you're not a government worker, and we do expect to see more people sick, and with the more people sick because of that, we would expect some to even die. So you could even say it's life and death, not just bank accounts. And y'all have been hearing about that, I'm sure, right? Not to mention getting on an airplane. When some fool behind you might have something in their shoe. No,
Oh my God.
no, I'm just, okay, that was a joke and funny. Yes.
So E. coli, like the subspecies, is the one that's very common.
Well, they're different subspecies of it. It's the ones that aren't found in your own. Every one of you in your colons, we have E. coli that is our friend, and it does wonderful things for us. It helps to make our vitamin Ks. It helps to finish off that digestive process that makes us healthy. That's the human E. coli. But then there are other serotypes that are found in other animals that can be deadly to us. And you know, cows, where do cows use the bathroom? We use the bathroom kind of in the toilet, whatever, we wash our hands. And that's our friendly E. coli anyway. But where do cows use the bathroom? In fields. And what does it do? Rains. And where does that rain take the cow manure and all the microbes that were in it? To the rivers, to the lakes, to the streams. And where do we get our water? From the rivers, from the lakes, from the streams, right? So, and that water is also used for irrigation of your spinach and your lettuce and your whatever you eat from the field. Casey's going back to macaroni and cheese. She doesn't want any of those things. So anyway, I'll boil them to death. So anyway, but you get what I'm saying. So this is natural, and this is natural, and it's okay to encounter a little bit of it, but if there's more of it than should be there, can it cause you to be sick? It definitely can. How many of you not heard of E. coli outbreaks? Because everybody's heard of those, haven't they? Well, that's where they came from, those things. And we have less of them because we usually have people monitoring and sampling and pulling things before they start causing epidemics. Salmonella is another one. So we're going to start adding to our list. So y'all have heard of things, haven't you? Listeria is the one, and I want you all to have this beside your notes. And Molly will get you in just a second. Don't forget it. I want you to have this one beside your notes. This is the reason that pregnant women are told not to eat deli meats. You know, don't go in the refrigerator and just pick up that piece of ham from the deli and just eat that ham or eat that hot dog or eat that whatever. This listeria is the reason, because it can cause spontaneous abortions. What do you all call spontaneous abortions?
Miscarriage.
Call them miscarriages, don't you? So this is why pregnant women, anybody remember being pregnant and being told not to eat deli meats? No, that's right, because I figured out most of y'all in here don't have any kids, right? But when you do get pregnant, good part of good prenatal care, they'll tell you some things that you should avoid that could be harmful to your fetus, right? And that's one of them. And it's because of listeria. That's very common. And they say, now it doesn't usually hurt anybody unless the numbers of it are high enough. Molly.
I went to like Yellow Packers the other day, and they said that like they had shut down and everything, and there were really low workers, and like the packages were disappearing, and they thought like not shut off food.
I didn't hear the first part. You went to mail a package.
Yes, ma'am.
Oh,
And they like advised against it because like there's social workers and like packages were getting lost and stolen and stuff.
stolen and yeah, right. And probably not really inspected. So think about those people who are sending things they shouldn't be sending, right? So that's another thing. It just affects everybody, doesn't it? So gosh, but we can't be afraid to live in our world. But we do, and things that you don't even expect. So thank you for adding that to it. I hadn't thought about that, but okay. All right, so guys, we're gonna start here. Because I have like no idea where we stopped, but these conversations are all part of the conversation. So I really like you bringing these things up. And look, Jenna really knows how to get me off on tangents. Come on, Jenna, with it.
Were you gonna follow up on the salmonella thing?
No, I'm not going to add salmonella to it. Our list yet.
She had asked, I think, so you're gonna follow up.
Oh, Amanda has brought up salmonella, and salmonella is one of those GI, gastrointestinal organisms that we are concerned about and you can get from food, but I'll add it to your list later. What I'm talking about, Michelle, is when I bring up an organism, you all and say, okay, now it's going to be fair game, you should be creating a list of organisms because they will always be fair game to be asked about on quizzes. And this way, if we do a few each time we meet, it won't be overwhelming when we get to them at the end, right? And the last thing in our class, the last day we meet, we'll have a final exam on just microorganisms, because that really is the most important thing that you all get out of this class, is that you become familiar with hearing their names, what diseases they cause, and what they are, so that you would know the right treatment for them. Correct? Correct? Okay. So we said we get that there are three domains. We have the eukaryotes, the bacteria, and the archaeobacteria. We get that bacteria and archaeobacteria are tiny. They are single-celled organisms, tiny little organisms that don't even have organelles. They don't have a nucleus. They don't have any of those specialized structures within. And then eukaryotes are much more complex. We get that. We know that. We also, I think, talked about viruses. Did we define why they weren't considered alive? Why are they not considered alive?
Active or inactive?
We don't call them alive or dead. We call them active or inactive. And we said because not even all viruses have their own DNA, do they? Some of them have DNA, but some of them just have RNA as the nucleic acid. We'll talk more about that in a little bit. But they cannot do anything on their own. They must be inside of a living host cell, before they can replicate themselves, before they can do anything. So this is why we talk about viruses being what they are. I want you all to know that when we think about cells and living things, we said the cell is the smallest thing that has unique properties of life, right? A cell does. And a virus isn't a cell, is it? It's not. So the cell having the smallest thing that has unique properties of life is one of the premises of the cell theory. And have we talked about that? Y'all got to remind me what we talked about in this class. Okay, so the cell theory is a scientific theory. And this theory states that The cell is the smallest thing that we know that has properties of life, and that all life comes from pre-existing cellular life. And I would want you all to know that Louis Pasteur is the scientist who finally put this to rest, that this is, or finally made it so that we accepted in the scientific community the cell theory. He's given credit for us getting to the cell theory, but he is also considered the father of microbiology. So Louis Pasteur. Louis Pasteur is considered the father of microbiology. Yeah, I know, right? Okay, so Louis Pasteur, and mostly knowing he's the father of microbiology. The experiment, his experiment, his final kind of experiment, after 400 years of experiments that were proving that cells are the smallest things that are alive, and that life only comes from pre-existing cellular life. After 400 years of repeated support of this, finally this is his last, well, not his last experiment, but this was the last experiment that sort of put the other theory to rest. The other theory that got put to rest and was disproved was the theory of spontaneous generation. So the cell theory replaced the theory of spontaneous generation, which stated, this theory of spontaneous generation stated that life came from non-existing life, that life could just spring up from non-life. And so anyway, this got disproven with finally, after, as I said, several hundred years of experiments, and finally Louis Pasteur's experiment that is now still a sterile vial of liquid in a museum, I think in London. So anyway, Louis Pasteur. Now, we talked about a microscope. If I go over the names and don't discuss the names, are y'all going to be okay with that? That you know that I don't need you to know them, right? This is just FYI. If I'm not giving them to you, then I don't really need you to know them. You guys have probably all heard of Joseph Lister, though, and Listerine. Yeah, Listerine, you've heard that. So these are people that were instrumental in us understanding the control of disease had to happen at this microscopic level. Sinowitz, this is a person who was really important because of hand washing and the importance of hand washing. Just the history of what he sort of went through was kind of fascinating, but anyway, hand washing being so important in helping to prevent the spread of disease. What do we know today is the single most important thing that we can do to help prevent the spread of disease? Hand washing is still considered to be the most important thing we can do, and you should know that is going to be hand washing. Are the little pumps that actually, are they the same as washing your hands? No, not the same at all. Actual hand washing is still considered to be the single most important thing we can do to help to prevent the spread. Now, the germ theory of disease, I do want you to know about Robert Koch. Robert Koch came up with some what were called postulates, which you really could just consider to be steps. And they call them logical steps, and they kind of are logical steps in helping to realize that disease is not caused because of the side of the railroad tracks you were born on, or the color of your skin, or your age, or what method you decide to have sex in, or who you decide to have. It's not. It's not caused because of any of that. Disease is caused because of cause and effect. Cause and effect. So Robert Koch was one of the first that gave us these steps to try to figure out what the etiologies of disease are. I want you to write down the word etiology because I'll say it quite frequently. E-T-I-O-L-O-G-Y, etiology. Etiology is just the study of the cause of disease. And I want to, you probably have heard of Robert Koch because of his studies with anthrax. How many of you have heard of anthrax? Everybody in this room should be raising your hands. I know you've heard of anthrax. And where have you heard of anthrax? First, let me give you the organism that causes it. It's a bacterium. So it's not a virus, it's not a fungi, it's not a protein. This is a bacterium. The bacterium is genus Bacillus. Bacillus with a B as in boy. Bacillus, species Anthracis. Anthracis. So this bacterium, Bacillus anthracis, causes a disease we call anthrax. Where have y'all heard of it?
The mail.
Oh, good, it's up there. You've heard of it in the mail? Okay, speaking of the mail.
They were sending it.
So because people send it for what type of activity?
To hurt people.
So terrorism, right? So to poison, to hurt people as a terrorist act. But Bacillus anthracis causes a disease called anthrax, which is has a really high mortality rate. Really high mortality rate. And you don't have to be young or old to die from it. It can be anybody, and it has a really high mortality rate. So, so this particular bacterium has been used as a bioweapon, right, in biowarfare. And when you hear about biowarfare, it means using something alive to initiate war, if you will, or to terrorize. So, absolutely. Robert Koch, though, this also causes disease of animals on farms, and it's not just limited to humans. It can cause disease that can be devastating to farm animals and to humans. And so he was trying to figure out what this, they knew what this disease, the signs and symptoms of this disease, but he's trying to figure out what causes it, right? So these postulates he came up with are still used today. And this is what I want you to realize. These are still used today when we are encountering an infectious disease, a microorganism. I'll go back to this a second. A microorganism that we don't, or disease that we don't know what's causing it. We still use his steps to figure out etiologies. So let me tell you these steps briefly because they are very, very logical. Are you ready? And you could briefly discuss these steps because they are still being used in our infectious disease labs around the world today to figure out diseases that, what might be causing signs and symptoms of particular diseases. The first step is to get specimens from something that's sick, an individual that's sick. So from someone that's sick, get isolates, if you will, specimens, and grow out a particular organism. So that's the first step. Does that sound logical? Okay. So the next one is to, and you guys, there's humans for this. Unless you're Hansen. Hansen used humans for this. But actually the next, they used to like test animals in the lab, mice. I don't know, there's some little mice. But anyway, the next step is to take healthy organisms, lab animals, and put this unknown, whether this is caused, so far it's unknown whether it really caused that initial thing to be sick. Put them in healthy organisms. The next step is to see if those healthy organisms got sick. And do you think that's the last step? Why not?
Because you have to figure out.
Because it could have just been coincidence that the things that you inoculated got sick, right? Mm-hmm. So then you have to isolate from the sick ones you inoculated and get that same organism out of them. Does that make sense? So after doing that, those are the steps that are used, and they're still used today. They are Koch's postulates. So Robert Koch is credited with something called the germ theory of disease. And the germ theory of disease says that all disease is actually caused by cause and effect. It's going to be some agent that's causing the disease. not because, you know, some supernatural thing doesn't like the way or who you had sex with, right? Or, you know, the color of your skin or whatever, right? Now, are there certain activities? So let me say this. When we find out etiologies of disease, the study of disease, and this is all disease, but the study of disease is called epidemiology. Epidemiology is the study of disease. So epidemiology is the study of disease, and it's everything about the disease. It's not only the etiology, but also signs and symptoms that are associated with it, how easy it is to transmit. Are all infectious diseases the same as far as ease to transmit? No, obviously not. Some things are really easy to transmit. They're airborne, aren't they? Some things, though, you'd have to have what? You'd have to have, like, it isn't even touch. You might have to have direct fluid contact. It might even have to be a specific fluid, right? So in epidemiology, for any disease that we know, epidemiologists study that disease, knowing not only the etiology of that disease, but they know how much you need to actually encounter before you get sick. There's some diseases that you can come in contact with just very few microorganisms and you're going to get sick. Other diseases, you have to have a significant inoculating dose. They call it an infectious dose, before you'd ever get sick. Let me give you an example. The common cold. Everybody's had a cold virus, haven't we? There are hundreds of cold viruses belonging to different families of viruses. For a cold virus, for you to get sick with a cold virus, you can swallow millions of them. So somebody can cough on your food, and it might disgust you if they did that. They could cough on your food, and you could swallow millions of them, and you're not going to get their cold. But if they coughed on their hand and touched this doorknob, and you touch this doorknob and rubbed your eye or your nose, one will cause you to be sick. One. So an epidemiologist, for all diseases that we talk about in this class, and we only get to talk about some of them, but all of them that we talk about, for all diseases, they're known etiologies. They know how much it takes to be an infectious dose. For all diseases, epidemiologists have given us the understanding of what the incubation time is for them. Do you think all diseases have the same incubation time? No. Some have a few hours, and some have days. For all diseases, and epidemiologists do this, they tell us not only how they're transmitted, what kind of incubation times we should be expecting, certainly what the cause of it is, but they can tell you also from these diseases whether you're infectious during that incubation time. For some diseases, you're infectious to other people all the way through the incubation, the time you're sick, your recovery time, and even during that recovery time, you can be infectious. Not for all of them, but for some of them. So an epidemiologist is studying all of that, and for all these things we study, these are known events or things, right? Everybody good with that? So Robert Koch gave us that. All right, so all kinds of new things are being discovered. Have y'all ever heard of biofilms? Biofilms. I kind of love this picture from your textbook. Do you see? Have y'all ever seen this picture? So this woman or androgynous being here, whatever you sort of saying, whatever. It's really that we're humans living in microbial gardens, right? We really are. And those gardens had better be just right. They better just look just right. By the way, healthy gardens, everybody's unique healthy gardens, they're different from other people's And here's what you need to know too. Families tend to have similar gardens, but would that make sense? Family members within one family have similar gardens. Would that make sense? Would it? Yeah, because they're usually being exposed mostly to the same things. But we know understanding someone's microbiome, and I want you to know this word, microbiome, means that you understand their microbial gardens. And by the way, they are as unique to an individual as fingerprints are. And it's really thought that in the very near future, somebody is going to be able to be recognized and identified by the organisms they left behind. Supposedly going into hotel rooms, so it might cause you to never want to go into a hotel room again. But supposedly going into hotel rooms, the people who have been there before you have left their microbiomes, some of their microbiomes behind, and they're very identifiable. They are to the individuals. So how fun is that to think about?
Delicious.
Delicious. Okay. So this human microbiome project, if you will, and I do want you to know the human microbiome project is just really a little bit of baby in its infancy. It's in its infancy, and understanding this and studying this. And if I had any money to invest, I'd invest it in this because I feel like we're just beginning to understand the important role in this and why we're seeing. I don't know if you all, Janet can maybe back me up on this a little bit, not that you're as old as I am, Janet, but I just know that you might be able to back me up on this. But you guys, we're really seeing since in just a few decades since I've left, well, many decades since I left like K-12 school or whatever, we're seeing way more incidences of things like autoimmune diseases. Like when I was in school, nobody had inhalers for asthma. We didn't have people who had hypersensitivity disorders. And certainly I knew one person growing up that had rheumatoid arthritis, but I knew no one who had Crohn's disease. How many of y'all know people with Crohn's disease? How many of y'all know people with lupus? How many of y'all know people with MS? I knew no one. There was no one. Those numbers of those diseases are going up, and they're going up at, fairly alarming rates, but what is probably playing a huge role in that is the change in microbiomes that are also happening. So understanding how and why an individual deals with disease is going to be, I think, just linked completely with their microbiomes and understanding their microbiomes. So anyway, I just want y'all to have heard of that. Now, as far as the macromolecules that make up cells, because we said cells are the tiniest things that we know that have properties of life, you all will probably remember these from other classes. I know some of you said you haven't had, like this might be your first biology class, but I'm just going to tell you what I want you to know from them. Inside of our cells, whether they are eukaryotic cells or whether they are prokaryotic cells, like the bacteria and archaeobacteria, there are four major macromolecules that are associated with life. And they are, you would want to know, carbohydrates, lipids, proteins, and nucleic acids. So those are the macromolecules that are associated with life. These are the molecules that contain a ton of carbon. And that carbon is that element that I told you all, or have told some of you all, that is that backbone element of all living organisms, carbon is. And it's because of just the unique properties of carbon that we know it will form long chains with other elements. It will form rings with other elements, and it will form these huge, which is what macro means, these huge molecules that end up taking on very definitive structures, and those structures will actually dictate how they function. So all cells are made of these four building blocks. Just like homes are made of cement, wood, I don't know what for homes, glass, whatever. These are the building materials for a cell. So these macromolecules. Now these big old molecules are actually made of smaller subunits. So when we think about smaller subunits, we call these monomers. What's another name that we call carbs? Can y'all tell me? What do we call carbs? Sugars. That's exactly right. And a monomer of these big carbohydrates are going to be monosaccharides, like glucose, right? Y'all know if you ever hear a suffix "-ose" or a root word "sacca," you're dealing with a sugar, which you know falls in what category? Carbohydrates. Exactly. What do we usually call lipids? Fats. Fats a good thing or a bad thing? Good thing. They're good things. They are the building materials of our cells. They get a bad name, don't they? They get a bad, just like carbs can be bad. They can be bad. Not all carbs are created equal. Some of them have empty calories and have no nutritional value, so those are not great ones. But lipids tend to get a really bad name, but they actually are necessary for building materials. And cells are essentially what kind of manufacturers? Proteins. So proteins is the most diverse group. And what do you all know? I want you to tell me and remember it. What is the monomer for proteins? What are the monomers for proteins? Amino acids. How many different amino acids do we have? 20 different amino acids that make up all of the proteins that we make, and we can make millions of different proteins. So using these different building blocks, just 20 of them, arranging them in a particular way, we can make all kinds of molecules that have a diverse function, right? Diverse function from each other. They're going to be amazing for us, right? So make sure that we kind of remember those basics. A function of carbs is not just as building materials for our cells, not just as building materials for our cells, but also as a major energy source for our cells. So we use carbohydrates as our, and I would want you to remember, our cleanest, quickest source of fuel for our cells. Cells are alive. That means that they're working 24/7, right? Do cells take breaks? No, that means they're working, they need energy. How cells best get energy are through carbohydrates. So we don't just build carbohydrates to be building materials like the roof on our houses. We also use them for energy, and that is a major component that we use them for. Delicious. What'd you say? Plus they're delicious, you're right. All right, so it's fine for now. And lipids, we said, are going to be building materials, but they're also, lipids can be used as a energy source, can't they? As an alternate energy source, not as good as carbohydrates as far as the cell goes, but as an alternate energy source, it can also be used. But you see this carbon as the backbone here for these triglycerides, these little fatty acid tails, you see? You've got carbon as the backbone. And whatever. So carbon is our is that element that is the backbone for all these organic. When you talk about organic chemistry, you are talking about molecules that are made from carbon that has a certain relationship with hydrogen. But that's what you're talking about, is a molecule of carbon that has joined with other elements, especially hydrogen, in a typical proportion. So most of them are huge, like carbs, lipids, proteins, and nucleic acids. That methane is also an organic molecule, and it's not huge, but anyway, they're usually huge. So organic versus inorganic chemistry, which is dealing with what? What would be a good rule of thumb? What would be a molecule that's inorganic that you know is necessary for life?
Water.
Thank you. It's H2O, because do you hear anything about a carbon in H2O? No. What are some other things that are going to be charged particles that are necessary for living systems because they provide electricity? They are called electrolytes. Another name for electrolytes are ions. Another name for electrolytes and ions are salts, like sodium chloride. Do you hear anything about a carbon? No. So electrolytes, though, ions that provide electricity for all living cells, they are inorganic, but are they necessary for life too? An inorganic molecule is one that really doesn't have carbon, which isn't exactly true because CO2 is what? Well, CO2 is actually a gas that is inorganic. So it has carbon, but it doesn't have hydrogen, does it? CO2.
So the definition of inorganic, it just means it does not have carbon.
Except for CO2. Yes. 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 Essentially, that's exactly right. So inorganic doesn't mean it's not necessary to life, right? They're just those molecules that don't have that carbon backbone, right? All right. And carbon is the defining one. Can you all see that? Oh, what? Okay. I don't know why they did that. All right, here it goes. So lipids, and lipids we really did say, lipids end up being building materials for our cells. Are y'all good? Mm-hmm. Okay, so lipids end up being building materials for our cells. They also end up being another source of energy that can be had. Proteins, those, they really are. These are 20 different amino acids that make up the monomers that come together to form a polymer. What does mono mean? What does poly mean? There's a prefix always. Multiple, right? So a polymer means a bigger molecule made of monomers, right? So this bigger molecule. And these proteins really have a very, very diverse function. Their structure dictates their what? Function. And I would want you to know that when you think about the structure of proteins, you usually tend to think about 4 different aspects of the structure. But the main thing I want you to know is the primary, because this is where it all begins. And what the primary structure of a protein means is this. Are you listening? It means what order did which amino acid come up and get in line? What order? Because it's like a beaded chain. So if you think about a beaded chain, like this little chain here, it means that there's an amino acid in this first position, there's one in the 12th position, there's one in the 87th position, the amino acid. You get what I'm saying? And the primary structure is dictated on which amino acid got in which spot. That can't be important, is it? It's crucially important which of the 20 amino acids have gotten in each of the spots, because that dictates all the rest, the second, the tertiary, and even the quaternary structure of the protein. And structure of the protein dictates its what? And are you telling me, Dakota, if glycine is just not right here, or valine and glycine switch spaces right there, that could affect the function of that protein? In what kind of a way? Like how? Not too bad of a way, right? Just, are you talking about out of this chain for this protein, this chain of amino acids, if glycine and valine just switch places, that would be such a big deal? It could be. It could be immediately fatal to the cell. In just one protein, just one chain, just one switch of amino acid, it can be lethal. It can be lethal, immediately lethal, or it can just be devastating. So what was the example I gave you in anatomy and physiology about the hemoglobin molecule that has 4 chains that are playing together, and one amino acid is out of line, out of hundreds? What was that disease? Sickle cell disease. That's what sickle cell disease is.
So that has 4 chains?
You don't need to know that. I was just telling you, look, Brandy, y'all, look, guys, y'all don't have to know everything. I was just saying that protein structures are important because the structures dictate the function. And that the structure is based on which amino acid comes up and gets in line where. How many different amino acids are there? 20. But in a chain of amino acids for one protein, you may have 100 plus more amino acids. The one that's in the 87th spot is important? Yes. Because if that amino acid is out of place, it could be devastating to that protein, and the protein could be devastating to the cell if it's not there, it's not functioning the way that it should. the example I gave was hemoglobin in humans. It's an inherited disorder where some people inherit the disorder where one amino acid in their hemoglobin is out of place. And it can cause a disease that's devastating with sickle cell anemia. So I was just trying to impress upon you about how important that is. All right, so that's proteins. But let me ask you something. When a cell is building its proteins, because a cell, that's what cells do. Y'all just told me that cells are protein factories. They're just making all kinds of proteins. Yes, they're making carbs. Yes, they're making lipids to be their building, but they're making proteins. And when they're making those proteins, what is dictating which one comes up and gets in line? The DNA dictates it. So that's our next fourth thing, is this idea about, it talks about anything I haven't said. I know a lot of this is repeat, but it's the nucleic acid, especially the DNA. It contains, when you hear that DNA is the signature molecule for living organisms, when you hear that, what you're really hearing is that DNA, deoxyribonucleic acid, holds all the recipes for all your proteins. And when I say recipes for proteins, all I'm telling you is that the recipe tells which amino acid when to come up and get in line. Right, Janet? Does that make sense to you? Does it make sense to everybody else in here? So DNA, that whole molecule holds all the recipes for all your traits. And when I say that, it just means the recipes for your characteristics or the recipes for your proteins. I could be interchanging those things. Recipes for proteins means that a recipe is telling which amino acid to come up and get in that spot, right? So that's what that does. The DNA molecule in all living organisms, the DNA molecule holds all the information. It is the whole cookbook. But when a protein gets made, it takes an individual recipe in the whole cookbook. So when you are making an individual recipe, like lasagna, okay, suppose you had lasagna you had to make. You had a cookbook. You don't have to make every recipe to make the lasagna, do you? To get to the lasagna, you open up just that recipe. You have it right here. This recipe will be written, right? And what that means is it will dictate which amino acid comes up and gets in line when. And when the chain is done, it'll dictate that structure of that protein. Is that right? That's DNA. But what helps it carry out that is that before it can do that, it actually transcribes that recipe to another molecule, and that little tinier molecule that holds one recipe is an RNA, a ribonucleic acid. So you can think about RNA, and there are different types of RNA. Yes, there are, and I know y'all remember them. But for right now, for us, you can just understand that ribonucleic acid is a much smaller little nucleic acid that is essentially helping the DNA molecule to write one recipe at a time. Is that right? Because you didn't have to cook your whole cookbook to get to your mama's lasagna, did you? One recipe at a time. Okay. We understand this DNA molecule is double-stranded, don't we? Don't we understand that? Anybody? Has everybody feel pretty familiar with DNA, the molecule? If I say anything you don't feel familiar with, because you haven't had, I know my A&P people better than remember this, but no, they are. It sounded like a threat. But if somebody's new to me that doesn't know what I'm talking about, stop me, okay? All right, so for now, I think, and this is all, I don't ever ask you what we'll talk about again in here. DNA is a double-stranded molecule, which means that there are these two strands that have these rungs, like a ladder, if you will, these rungs in between, that are then twisted, right? This is what this molecule looks like. And you would think, since it holds all the recipes for all your traits, that it would be an incredibly complex molecule, but it is not. It isn't. And not only is it not complex in our cells, it's not complex in any bacteria, bacteria, proteins, fungi, animal kingdom, this molecule looks the same. Now, what causes the diversity that you see between single simple life form cells and complex life form cells is the amount that you have, is the numbers. But the DNA molecule itself is exactly the same. It's actually, when you hear about the uniformity of life, it's referring to this molecule that is holding every recipe for everything that will ever happen in a cell. This molecule is incredibly simple, and it's not different between the bacteria and the archaea and the eukaryotes. It's not different. Its structure is identical. The unity and uniformity of life is really true at this level. This DNA molecule is incredibly simple. The strands have a, I'm not pharmacy, but a phosphate, a deoxyribose, which is a sugar, phosphate, deoxyribose, phosphate, deoxyribose. Does it change? Does it change? No, it's the same. You mean in animals? But how about in fungi? Same. How about in bacteria? Same. Strands are the same. Well, the diversity must come when the rungs, right? 4 nitrogenous bases. 4. Adenine, thymine, cytosine, and guanine. Yes.
Don't viruses have DNA as well?
Some do. Some viruses have DNA, but they don't have any of the complementary things that are needed for movement, for division, for anything else. Some have little pieces of DNA, but some don't. And some of the most deadly viruses don't. They just have an RNA. All right. So, so those 4 bases, guys, you ready? Adenine, thymine, cytosine, and guanine. Correct? No, but you can look them up. They're right in your book. Adenine, thymine, cytosine, and guanine, right? Okay. And then the RNA molecule, ribonucleic acid. These are typically the ones we think about that are called messenger RNA molecules. They are single-stranded, and they're much smaller. They're much shorter because they're just going to typically hold one recipe for one trait. So that's kind of what I want you to know now. Does anybody have any questions about that, what I've said? Okay, now, ATP. This is called the energy molecule. I know you guys have heard of this as being the energy molecule of the cell. It stands for, you should know what these things stand for: DNA, RNA, and ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate. And it's composed of adenine, ribose. What is ribose? How would I know what it is because of its suffix? It's a sugar, isn't it? And it's got 3 triphosphate molecules. So anyway, and all together, when they're acting all together, this molecule is called adenosine triphosphate. Adenosine triphosphate. And I really do like the structural formula for this molecule to look at. And I'm no way an organic chemist or love organic chemistry, but I do just kind of love this molecule because it is the structure of this molecule that helps us understand how it acts the way it does. Yes?
What was the third composition?
It has 3 phosphates, triphosphate, right? 3 phosphates. So adenine, ribose, and 3 phosphates is called adenosine triphosphate. This adenosine group is going to be the sugar and this nitrogenous base here, adenine. So these little 3 phosphate groups that are held together, held on to this almost like the tail of a kite, isn't it? If you think about this as being the kite, this is the tail of the kite, this molecule. The bonds, the chemical bonds of this molecule, the chemical bonds of these, this is, and I'm the tail, I'm the last phosphate group, go ahead and hold on. First phosphate, second phosphate, I'm the last phosphate, and there is that adenosine up there. This chemical bond, there's energy, try to get away from me. Do you feel the energy? There's a chemical bond here. There's an energy, and this, she can ask Jake, can't beat me in my arm wrestle, can you do it? It's genetic. It's genetic in our family. We don't get beaten in our arm wrestling. So mine. So anyway, no, but in chemical bonds, there's energy that's just like that feels just like that, right? And what that energy is really like a stored battery of energy. But what can happen in this particular one is it can break away. And when it does, it's going to give off energy, just like a battery. Y'all can break off, because now it's adenosine diphosphate. Now I didn't get destroyed, but when I let go right here, it powered something else to happen in the cell. Energy was released from this site. Energy was released, right? And it drove something else to happen in the cell. I'm still here, they're still here, adenine diphosphate. And if a little bit of energy is put back in, guess what I'll do? I'll recharge. Now I'm recharged, I'm ready to give off more energy for something else to happen. Then I'll recharge, that battery will recharge, and then it will give off energy for something. You get what I'm saying? It's in the old one, right? Are you good with this? So this molecule is considered the energy molecule of cells because it is beautiful in its structure that it does that. It can act like ATP is a charged battery. And just like batteries drive reactions in your appliances and whatever you use, they do the same thing inside your cells. This is a charged battery. And then it can also be readily recharged. Are y'all good with that? All right. Now, this is something I feel like I can't believe we haven't gone here yet, but okay, that's fine. I'm good. Taxonomy. I thought we'd be in chapter 2 by now. So, okay, it's all right. Taxonomy. This is the science of classifying things. You know, scientists sometimes are given a bad name because they're like, oh my God, y'all just like to label and classify everything. But really, it's very important to do this, and it's not just important. It's not just important because of like OCD reasons. It's important because of the disbursement of information across even languages, right? So this is going to be really important. You'll hear Carl Linnaeus' name. You'll hear Carlos Linnaeus. You'll hear Carl Linné, whatever. What do I want y'all to know is I've always referred to him as Carl Linnaeus. But anyway, just write it down like this since it's in your notes. It's fine. He's given credit for this this taxonomy, classifying things with a nomenclature of 2 names. It's called a binomial nomenclature. Binomial meaning how many names? 2. And what do y'all already know about the first one? It's called the what? And the second? Species. And the genus, can you have many species? Sure you can. Okay, so we know that. So assigning scientific names, and this by doing this, no matter what language you might speak, whether it's a foreign, another language, whatever, the disbursement of the information is just quicker this way, so everybody's kind of on the same page with this. I must be hitting this somehow when I walk back. All right, so classifying this nomenclature. I do want you all to know how to write organisms' names. The genus is always, and here's your Escherichia, the genus is always uppercase, right? And the species is always what? Lowercase, right? If I were to ask you and give you some examples on a multiple choice, which one is correct, you would know how to do it. Or if I were to give you just a name with nothing capitalized or whatever, you would know how to write it. You would know that it's either going to be italicized if you're using a computer, or you're going to underline it if you're writing it. Isn't that right? And you'll know you're talking about the actual name of a living organism, the genus and species. Classification, I do want you to have the basics of this and understanding this. We said we have 3 domains. How many kingdoms do we have? 6. And under each kingdom, there are what? The phylum or phyla. Then under a phylum, there's class, order, family, and then genus and species. What we will deal with in here are genus and species. That's what we will deal with in here. Okay? All right, so I kind of just want you to know, like under bacteria, I'm not for bacterium, the kingdom, bacterium. I'm not going to want you all to know family, order, class, we're just going to talk about genus and species, but I do want you to understand this. So you understand if you're in the same species, I just told you all today, I hinted at this today, I said that you can actually be in the same species, but there be different serotypes of it, right? Which organism was I talking about when I talked about that? E. coli. Exactly. So you are really, really related, aren't you? There's just slight differences in products that you actually secrete or possess, or properties that you have that have made you one of the serotypes. So anyway, does everybody understand this whole idea? Okay. So, and they give you some examples. Humans, what do we belong to? What do humans belong to? What genus is Homo? And what species? Sapiens, right? So Homo sapiens. Are there any other species within our genus? No. No. Okay. Phylogeny, and this is important, this definition to kind of know, this is the scheme that shows you the relatedness between species. So this is this idea that it shows you the relatedness between species, because some species are more closely related than other species. And this is not just at a macroscopic level that you would expect, but this is actually done in biochemistry as well, as far as the proteins, that the DNA, the recipe for the proteins that species are making, how closely they are related. So again, not just at a macroscopic level, but also a biochemical level as well. In evolution, we sort of talked about evolution and survival and through natural selection. This has nothing to do with individuals, is it? This just happens to be that the traits in that period of time, the percentage of traits that you see are going to be the traits that help that species to survive. So in the next generation, you tend to see a little few more of them because they got to survive, right? So an adaptive trait is a trait. An adaptive trait just happens to be a trait that is going to help you to survive that particular environment, right? I'm not going to talk about that one. And this is just looking at some of the schemes that you can use. So any questions for me about this? Guys, I want to start. I know I only get a few more minutes, but can I have a few minutes just to introduce something? I want just a couple minutes to introduce it. And I want you all to be familiar with this. There are what we refer to as the 5 eyes of microbiology. 5 eyes of microbiology. We said that there is nothing as devastating to the human body, as rapidly devastating to a human body as an infectious process. Do you remember me saying that? There's no metabolic disorder, there's no whatever that's going to hit you today and take you out of this world tonight. Not even any cancers or anything like that could do that quickly, right? But infectious diseases can. The problem we have is that to properly treat, because if you're not giving the right treatment, what could you be doing? Harming. That's exactly right. To get the right treatment, we have to know what we're dealing with. And oftentimes, that's tricky because signs and symptoms can overlap, can't they? I've had students in microbiology who I just hear them sometimes say, flu-like symptoms. Because what are flu-like symptoms attributed to? A ton of things, right? A ton of things, not just flu, are they? Influenza, but a ton of things. And it's really true, and it can be really, really tricky. So in infectious disease, we realize that this person coming in with a really high fever, and maybe their flu-like symptoms, I don't know, but really high fever, whatever, could have something that could take them out of this world in quick hurry. But we've got to get to the right answer as quickly as possible. And unfortunately, to get to the definitive diagnosis, which you all know the difference in definitive versus presumptive, it's all a lot of different spellings of that. Definitive diagnosis, we have to actually have that microorganism we're looking at and that we're growing. And can it take some time for that? It can take time. These are what we have to do. These are the steps in microbiology to get to the definitive diagnosis. So I want you to know them. They actually make good sense, you know, as far as common sense goes. Inoculation, incubation, isolation, inspecting what you've isolated to get to your what? Identification. So inoculation is, and I just want to do it briefly, and then I'm going to let you guys go, so if you'll bear with me. Inoculation, and we'll talk more in detail, but this means getting the specimen that you need inoculated onto media that you know that your potential pathogens can grow. Do we have a definition for pathogen? Something has potential to cause disease, right? So inoculation, you've got to have the specimen you need and inoculate it onto the media that you need that could potentially grow it. Then you have to incubate it, which means you've got to let it grow, right? Incubation is going to take things like temperatures. It's going to certainly take time, but temperatures, certain temperatures, and also certain atmospheres to incubate it so that this whatever you inoculated, if the pathogen's there, it's going to be happy. And it's going to start under that incubation time starting to divide, right? After the incubation period, you've got to isolate them out. Because do you think that when you get a specimen, it's not going to be contaminated with normal flora? No, it likely is going to have a lot of things in there growing with it, right? But you have to recognize what might be bad and isolate it out. From the isolation, you've got to inspect and do biochemical testing that will get you to the what? Does this sound like this is going to take some time? Yes. Wait a minute, you got somebody that was really, really sick. What are you going to do in the meantime? You're going to make them wait? What'd you say?
You can treat that for what you think it might be.
You're usually going to start treatment on the presumptive diagnosis. And usually that treatment is going to have to be what's called broad spectrum treatment that could hit a variety of things. And then as soon as you get the identification, what can you do to your treatment?
Narrow it down.
Does that make sense? So broad spectrum treatment versus narrow spectrum treatment. What's the worry about broad spectrum treatment when you're up here not knowing what you're dealing with? The broad spectrum treatment is usually more damaging to the person because you're having to use a drug that's going to be able to hit a lot of things, including maybe being dangerous to the person before you could narrow it down. You might even be not knowing what you're dealing with and dealing with something totally wrong, right? So in microbiology, the catch-22 is that we're dealing with things that can be the most devastating to humans that we know, but our hands are tied until we can get the identification, right? I'm going to tell you in this class about some things that we can do to narrow the time and feel more comfortable, right? But these are the 5 eyes of microbiology. And let me say something that's to you guys as primary care providers, because y'all know that I think nurses are primary care providers. Not doctors, nurses are, because they're the ones taking specimens, they're the ones doing what they need to do, delivering meds. They're the ones that are hands-on, primary care providers. Here's what I need for you to remember in your head. This identification can only be, you can only be confident about this from the integrity of the specimen that you took, you received. If somebody doesn't know how to get the right specimen, this is worthless. Are you good with that? That should be stenciled. Anybody who ever has to get a throat swab, a urine culture, a wound culture, a sputum culture from a Lucan tube, anybody who's required to get specimens from patients, I don't care if it's fecal specimens, they should have that embroidered somewhere on the wall that they understand that the ID that's coming out of that is only going to be as good as the specimen you sent. Are you good? Are we good? So it's going to start with what we can inoculate, right? So anyway, you'll have a basis for what those eyes are. what the eyes of microbiology are, okay? Good? Yep. All right, so I will see you Wednesday. You guys will be ready for a lecture quiz on what we talked. Did we talk about anything today, Brittany? Did we did? Okay. So we'll have a lecture quiz on Wednesday, first thing, but also, let me say this, let's go ahead and let's just do our little microscope at the beginning of lecture 2. Because are there any questions for me about the microscope labeling? Do you all have that down? Do you also know the answers to the 2 questions about microscope?
So 2 quizzes on Wednesday
Yeah, just 2, a lab quiz and a lecture quiz.
at the beginning of lecture.
At the beginning of lecture, both of them. And remember, any microbe we've had, should you be familiar with it? Yes. So any microorganism we've had, you've started your list. Any questions for me before I stop the recording? No? Okay, see ya.
Your first one?